Control deck, that is Hercules away.
Copy, Hercules away.
Control deck, that is Hercules past the transom.
Copy, Herc is past the transom.
Control deck, that is tether all out.
Copy, tether is fully deployed.
Control deck, can you have Hercules pull forward just slightly, please?
Roger, pulling forward.
Control deck, that's Atalanta in the water.
Copy, Atalanta is in the water.
the gauges up. I'll go to dive salvo in a minute. I'll do it now. Oh wait, we want new dives. Let's do that one. Where's gauges? I got switched. Oh. Oh, nice. Okay. Perfect.
This is an audio slate for dive H2069. UTC time is 05:22:10.
Control, back deck, all stop at 50 meters. Control is being sent to the fan.
Copy, all stop at 50 meters.
Ready to give it a bump? Go for it. Confirm control. Alright, we can start heading down. It's alright, DC ground. When you get that settled, can you... Oh wait, Adelaide's lights are on. That should be fine. Okay. Oh, you know what it is? It's the temp probe. The macro turns it on automatically. So yep, it's back to normal. It's good. All right. Yep.
Are the lights on Atalanta on? It's really dark. Here, maybe just... I don't know. I just made it auto. I don't understand it. Sometimes it just needs the handshake again. Yeah. So I just quickly made it auto and then I made it manual again. Cam port recall, yeah. So, want to look at that camera. And then cam starboard as well. Little changes. And then remembering to go back to the port camera on the controller. We can do it now. Yeah, when we hit that 50 meters, it's a good time. So that's in the software here. Yeah, so what we're doing is the SAT3, we're going to change it to that 4
Yeah, I turned them on.
box. So that is, remember, the head 5 and the 4, yes. And then, yeah, PIP 1's going to be your ROV Zeus.
Okay. Makes sense.
You can do cam rear or something like that. Yeah. Yeah, snap. You might want to preview what you're changing as well. All right. And you'll probably want to start getting ready to do audio levels for everybody. Yes. And then just go through and double check that. And now that we're kind of here, it's a good time to switch off. What do you think? Yeah. Yeah. Cool. Yeah, so handover is here. You would do your, oh, you didn't really do a system health check, but you should. So do that, and then you'll do a handover with Aaron. Because we haven't looked at the files yet, which we want to do. No. Arrow, yes. Hercules. And this looks, yeah, 26.9. No, you have to click it. And Hercules. Yeah, so it's ProRes H.264 Hercules, ProRes H.264 Atalanta. And what we're looking for here is that the files are growing and you want to do your date modified. Click that so that the newest one is on the top. Right now there it is, perfect. So then do that for all of them. So we can, that's this button here, and that should just give you kind of, go ahead, you can push it, you're not going to break anything. That's going to give you the recorders, it's going to give you your closed captioning, the vehicle cameras, which shows we have 2 black squares here. We need to remind them to turn on those cameras. Those are Herc cameras, the ROV upper and ROV lower.
Huh? No, go for it.
That's correct.
Yep, 100. Yeah, kind of just base it off the line there on the comm.
All right, good afternoon, everyone. Thank you for tuning in to nautiluslive.org. We have just launched the vehicles in our first dive into the Palau National Marine Sanctuary. For today, I'd like to go around the room, or at least start with the back row, if we could do introductions. So my name is yyyy-mm-dd. I am a Palauan, a science communication fellow for this expedition. Next to me is our science lead.
Thank you, Made. So Daniel Wagner here is watch lead and also sailing as expedition leader.
Hi, good afternoon, everybody. My name is Maikani Andres. I will be the scientist for this shift, and it's nice to meet you all.
Hi everyone, my name is May Lubetkin, and I'm the science manager on the ship and the data logger on this watch. Looking forward to it.
Thank you, everyone, for the introductions. So, Marang, Maikani, do you mind telling us a little bit about this dive that we are about to see?
Sure, I can start it off and then Maikani can fill in here. So we are just inside the Palau National Marine Sanctuary. Closest known feature here is about 45 nautical miles to our south, Velasco Reef, a drowned coral reef atoll whose shallowest portion is about 50 meters. And we are on a seamount that has no name, and that was until a few hours never had been mapped. So we just mapped it over the last couple hours, and it's a very interesting looking feature. The summit is about 1,000 meters, and then today the dive is going to land on the seafloor, acquire bottom at about 1,500 meters on one of the flanks of the seamount, and then we're going to transverse some interesting terrain going across about 500 meters of vertical relief, crossing various different habitat types, and then the plan is to reach the summit about 14 hours later. This is part of the first of 2 expeditions that are really trying to, for the first time, survey the Palau National Marine Sanctuary and a wide variety of habitats across it. So very excited to, for the first time, see these habitats and share it all with you.
Thank you for that, Daniel. So for everybody that's tuning in for the first time, if you do not know, Palau is a very small country located in the Western Pacific. It is composed of 16 states, Kayang being the northernmost state. And where we are right now, as Daniel mentioned, is around 45 nautical miles north of Velasco. So Velasco is a reef that is owned by Kayang State. Velasco got its name because of a ship that had sunk there before. And so, again, this is completely new area for mapping. We spent roughly 6 hours earlier today mapping the reef, and now we have launched the vehicles down. So Daniel, how long does it take for the ROV Hercules to reach the bottom?
Good question. So we descend typically at an average rate about 30 meters per minute. So for this dive, 1,500 meters, so that's just under an hour of descent. But there's several times where we stop. So for those who are new to our operations, typically there's a longer stop at around 50 meters for our front row to do a whole series of different checks. So we should be acquiring bottom probably a little bit after 3 o'clock local time. So very excited.
Yes, thank you for that. There you have it. We'll be hopefully around 3 p.m. local time, and then we will see the bottom.
Would it be possible to do some intros for the back row just so I can do audio checks?
Okay. And so again, I'd like to ask the back row if we could do another round of introductions to all our new listeners.
Sorry, I was not on the chair, so I need to do it.
So my name is yyyy-mm-dd. I am a Palawan Science Communication Fellow for this expedition.
Ali, aloha, good afternoon everyone. Daniel Wagner is sitting as a watch lead and also sailing as expedition leader on this expedition.
Good afternoon, everyone. My name is Maikane Andres, also Palawan, and I will be serving as a scientist for this watch.
Hello everyone, I'm May Lubekian, and I'm the science manager on the ship and the data logger on this watch.
Amazing, thank you.
Is it okay for the front row to do introductions?
Yeah, we're ready.
Yep, front row's ready.
Let's do it.
Yeah, in your front row, this is Megan Putz. I am your navigator.
And I am Jake Bonney. I'm sitting in the Herc seat.
So now if I could ask the front row to do some introductions, so starting with the navigator, please.
Okay.
Yep. So I'm Megan Putz, the navigator.
Megan, sorry, I don't think your mic was on.
It's on,
Are you on SPL?
and I'm on SPL.
Okay.
Can you not hear me?
Yes, no, perfect. Thank you.
Okay.
Yeah, go ahead.
Yes.
You're pretty quiet. Let me get you up there. And I'm Erin Rainey. I am the video engineer on this watch.
Thank you everyone for those introductions.
It's 12 to 4 watch.
12 to 4.
Yep, I can hear you.
Great. tomorrow's Halloween. So our next watch will be Halloween. Bringing it at midnight.
I heard there's a onesie in store for you.
Oh yeah, I'm so excited. I'm ready.
I am very happy to hear you have costumes prepared.
Mm-hmm.
As your SCF, I will be taking your pictures for tonight's watch. And those will be on the Nautilus social media account, so make sure to follow Nautilus Live on Instagram and Facebook and Twitter to get all the details and that we do not see on the live feed.
I've been so excited about my costume ever since the last cruise, and I decided on it. We had like I was on 12 to 4, and so we had like this whole side conversation about what we would be. Do you have a costume, Jake?
I do not yet, but
I think you can come up with
probably
something.
Scrounge up something.
Something that goes with your mustache.
Oh, okay.
I'm going to need some help too. I don't have one.
Waypoint 1, and then we're moving.
And then we're going to move sort of north,
Okay.
east-ish.
About 30 or so minutes until bottom.
Yeah.
40 minutes.
So when we're on bottom, are we gonna trigger a Niskin? Will that be one of the first scientific objectives?
I thought we were doing all the Niskins at one location.
That's only if we find something biologically interesting. We have interval depths, and one of which is bottom.
Okay.
Yeah. I also thought we were doing it before we ascend to the surface.
I thought that if we didn't find something interesting to trigger all the Niskins at, that we would do interval depths on recovery.
Yeah.
That's
that's the plan, yeah.
Okay.
So if you don't see anything interesting, we'll trigger one before we ascend, and then at the interval depths going up.
Okay.
Yeah.
Okay.
So basically, do our dive. If there is a high diversity community, trigger all 6. If we do not see it for the 14 hours, then when we leave bottom, probably about 1,000 meters, we would trigger one there, and then on our intervals, and that would give us a couple extra bottles, and we would just double up on some of the depths coming up.
Mm-hmm.
Okay, sounds good.
Oh, and the thing about the Niskins, May, is that
I know that the last two.
the
can only
last
be,
two.
Yeah.
So we start with 1 and go to 6.
Which makes perfect sense.
Right?
Numerically speaking as well, so.
Well, we had some issues on the last time where, I don't know, people just were really keen to start with like 5 and 6. But,
well, yeah, we're all briefed on that, so
sweet.
no problem.
Yeah, it worked out.
And while we're waiting here and descending, so our depth is currently 700 meters, so another 800 meters to go. Maybe if I can just ask Maikani to tell us a little bit about the environmental DNA monitoring program that Palau International Coral Reef Center has been working on for the last couple of years, and how these samples that we're collecting are supporting that effort.
Yeah, thanks for that, Daniel. So Pickwick has been conducting the eDNA monitoring program since 2021, I believe. Since then, we've collected about 4 times around PNMS within the artisanal and the domestic fishing zones. We've also done some ashore. And this program helps us determine what's within Palau's waters. That way we know the resources and the biodiversity within our water. So yeah, thank you.
A follow-up question on that, Maikani. So how many Niskin bottles are on the ROV right now?
So we have 6 Niskin bottles. The plan is if we see something super cool, super diverse on the bottom of PNMS, we'll trigger all 6. If not, we will trigger 1 at the bottom and then all 5 at interval depths coming up to the surface. We also do surface collections at each site that we go to. So right now, Piera, the other scientist with me, is, I think she's filtering right now. She's probably done with collections. So she's doing the surface samples right now.
And just to add a little bit of context, yeah, and just also our gratitude here for the Ocean Exploration Trust. So we started really planning for this expedition about 2 years ago, and then ramping up, and the Palau International Coral Reef Center has been an integral and critical partner in those efforts. And so we're really thankful for that, and really excited too to also be able to support collection of samples to support that long-term eDNA monitoring program, collecting not just samples at their regular monitoring sites at the surface, but expanding that too, collecting samples at depth to see how those compare. And then not just those environmental DNA samples, but also being able to collect visual information with our remotely operated vehicle cameras, as well as physical samples to correlate what we might see in the eDNA with what actual animals live on the bottom. So really thankful for this collaboration and what we're going to be experiencing, surveying and documenting here over the next few weeks.
Thank you, Daniel. I see a comment that has come in. Someone says, Ali and Ngilswelb. Ali in our local language means hello, and Ngilswelb means good afternoon. So the person is sending well wishes to everyone aboard Nautilus, and they are looking forward to watching and learning about the dive today. So thank you for tuning in. I would like to also encourage more Palauans to stay tuned in and send in those questions. So we would love to answer them on this watch. Thank you.
Absolutely. I'll just echo that one more time there, Imade. So the Palau National Marine Sanctuary is such a special place and a very ambitious conservation effort, yet is offshore and far away, so out of sight and hopefully not out of mind, out of many people. And when you can't bring all the people to this extraordinary place, our mission here is to bring the place to the people via tidal presence, via these videos. So hopefully you all can join us. We'll be live streaming here through November 22nd. Most days having dives exploring a wide range of habitats across the Palau National Marine Sanctuary, including various seamounts, underwater canyons, slope habitats, and a variety of others. So stay tuned, and hopefully you can join us as much as possible and share in the excitement of discovery and exploration.
Exactly. In fact, tonight at 6:30 p.m. there is a community ship-to-shore interaction with EV Nautilus. It is at Barracuda restaurant that is right next to Fish and Fins in Long Island. Pickrick is behind the organizing the event, and so there will be refreshments made available for everybody to go. So again, if you are local or a local resident and listening in, please stop by tonight at 6:30 p.m. at Barracuda and join the ship-to-shore interaction with the team on board.
currently at a depth of just approaching 900 meters, so about 600 meters to go, about 20 minutes till we reach the seafloor.
So again, to all our new listeners, we are roughly 45 miles north of Velasco Reef. Velasco, again, is known for its abundance of fish. It is a very popular fishing ground for local fishermen. And so it will be very interesting in the next 20 minutes to see what actually is in the bottom of the reef.
Yep. Norbit. I just power cycled.
Maybe taking the opportunity to add a little bit more context about today's dive. So we're diving on a seamount, which is another term for an underwater mountain. This seamount has a depth of about 1,000 meters. Seamounts are very important globally because they are known as hotspots of biodiversity in an otherwise typically somewhat less sparsely populated deep ocean. So seamounts are some of the most biodiverse and productive habitats that we have in the deep sea, but not every seamount is created equal. The shape, size, steepness of these features dictate what kind of biodiversity you expect to find. This one has a very interesting topography and shape, including some very steep areas. And in general, seamounts, they basically alter the current regime around them. They interact with the predominant currents to create areas where you have currents accelerating and flowing around them and creating favorable habitat for a wide diversity of organisms. And so, yeah, we chose this dive track to transverse some of these interesting areas. And the other thing that is interesting about the seamount is gets to have been 1,000 meters of the sea surface. So it intersects the depth range where on the open water, where a lot of organisms undergo diel vertical migrations. So in open water, there's a whole range of organisms, mobile organisms that go up and down the water column on a daily basis. That typically starts at around 1,000 and goes to the surface. So this seamount intersects that area. So it is an area where we expect to find more life than in other places of the deep sea.
Are there any particular organisms that you're excited about seeing?
Yeah, great question. I'm more of a generalist. I like to see the forests, those places where you have a lot of different things. And so yeah, hopefully we'll find some patches where there's a lot of abundant and diverse life.
I would really like to see the different types of fish down there. Very curious to see if we have any Dumbo octopus in particular. I've seen those on EV Nautilus channels, and they are very cute, and I would like to know if we have any cute animals down in our waters.
Oh, they'll be cute animals. I can promise that. Of course, that really depends on your definition.
What are you looking forward to see, May?
I'm looking forward to seeing the change over the depths, and yeah, I think I kind of can echo what Daniel said, seeing how all these different factors interact, the currents, the geology, the structure of the site, and then the corals I think will be interesting if there's corals and sponges and fish. We don't know what's here, so I think just getting a good general overview is what most excites me.
Yeah, here in Palau, the deepest trench areas have been explored and our shallow reefs have been explored, but the depths we're going to today have not been explored. So everything we see today is going to be new, which is really exciting. It's the best part.
How close were the Nat Geo Pristine Seas cameras to where we are?
They were more close inshore
Okay.
because their ship, the Argo, can't go offshore.
Oh, okay.
But around here, especially in island nations, you can find the deep sea just a couple miles offshore. You don't have to go very far. But yeah, going out to seamounts this far away, it becomes a little less protected, and it's challenging for a ship that doesn't have a DP system like we do.
Maybe just adding a little bit of context. So yeah, this seamount is a completely unexplored feature. As I mentioned earlier, this area had not been mapped ever before. So we mapped it this morning, and now are going to be the first times to dive on it. The closest ever historical dives of this area happen about 25 nautical miles to our southeast, where in the 2013, Jamstec conducted a series of dives using their human-operated vehicle or manned submersible Shinkai 6500. So there have been a series of efforts, as Megan alluded to, including the most recent one by National Geographic Pristine Seas, that did a series of dives and baited cameras, and over the last year or so, we've been working closely with various partners, including our expedition science leads that you'll meet on the subsequent watches here, Brian Kennedy and Jeff Drazen, and just gathering all that data and seeing what's been done before us, and then using that to plan our surveys, trying to fill in as many gaps as possible. There are just so many gaps yet, so over 70% of the seafloor of Palau has never been mapped with modern technologies. And so much left to explore, so much left to discover, and so excited to do it with all of you.
Yeah, the Nat Geo drop beta camera deployments had a lot of really cool sharks and fish that were attracted to them, and I'm excited to see if we'll be able to see some of those same species in our ROV dives. Unfortunately, we want to use multiple different types of sampling when it comes to the deep ocean, because the animals react differently to different types of sampling efforts. Baited cameras attract your predators and scavengers, where those same animals might be deterred by a loud ROV coming down to view the seafloor. So ROVs are really great at censusing our sessile animals like corals and sponges and other small invertebrates. When it comes to our larger, more charismatic fauna, those animals might either be attracted to the ROV out of curiosity or avoid it altogether because it is an unknown possible predator. It's not, but they don't know that. We're like an alien to them, you know?
Here is a question that just came in through the feed. What depth are you diving to?
The depth of this site is 1545 meters.
Yes, and then right now we're around 1,200 meters. So just in a couple more minutes and then we'll reach the bottom.
Almost there.
Almost there.
And while we're waiting, I also like to take the opportunity to acknowledge another very important person in this expedition team, whose voice you will not hear on the live stream, but has been actively participating in planning and executing the survey. So Ikelau Otto from the Palau International Coral Reef Center, who helped plan these dives, is also joining us over the chat, was joining us yesterday at midnight during the dive that we unfortunately did not reach the seafloor. So thank you so much, Ikelau, and I'm looking forward to continue working with you over the next several weeks.
So Irlau has been getting major FOMO for not being on the ship. So if she's listening, we'll send a bunch of pictures so you can just live on the ship as much as we are.
Oh my God.
And while we're waiting here, just a little under 10 minutes to reach seafloor, just to give you a little bit of a look ahead. So yeah, we expect to reach the seafloor here in about less than 10 minutes. Then it's going to be a 14-hour dive, so exploring the seamount till 4 a.m. Palau local time. But that's just the beginning of a very exciting expedition that we have for you. After recovering the vehicles tomorrow at 4. We'll be transiting further north to explore yet another unexplored and unnamed seamount, diving there probably just before lunch and doing a 12-hour dive there, and continuing this pace of moving on to a new unexplored feature, and then doing 12 to 14-hour dives, and that's going to continue almost daily through November 22nd, and I hope you can all join us for as much as possible. And also while waiting, take a second here to acknowledge our scientists ashore. So in addition to the 50 people or so that are on EV Nautilus currently, we also have a group, an extensive group of scientists, engineers, cultural knowledge holders, and others that are joining us remotely via telepresence technology. Currently in the dive, I already acknowledged Ikelo Oto, our chief science lead on shore. We also have Sako Matsumoto from the Chiba Institute of Technology in Japan, Dr. Christopher Kelly, formerly at the University of Hawaii, Jonathan Womack, independent researcher and historian. So thank you so much for joining us.
Dan might be messing with it. Yeah. Yep.
So just over 100 meters to go before we acquire bottom at about 1,550 meters.
And little bits of beams there.
Also want a big shout out to our heroes this morning, our deck team and ROV team who worked relentlessly throughout the night and the morning to fix various issues that we had on the ROV and on the A-frame and making this dive possible. So thank you so much for all the hard work and dedication.
Okay, so a comment just came in. It says, Ali, I am a Palawan living in and tuning in from Georgia. And the question is there a bait on the ROVs to attract the animals when we reach the bottom?
That's a really good question there. So we don't have a bait in the sense that people typically think of bait, so we're not carrying fish or chum or fish oil or anything like that. However, the ROVs themselves emit lights and electromagnetic signals, and that in itself can be an attractant for some organisms, but a deterrent to others. So yeah, Megan Putz earlier alluded to that there's different technologies and they all have different strengths and weaknesses. And yeah, so the lights, and that's an interesting thing that has not yet been quantitatively investigated in a comprehensive way, but some of the sensors do have effects on the animal in terms of attracting them.
Okay, we can continue on. We're going to go a little bit, we'll just slowly descend to the bottom from here. Yeah, maybe 20. Steel bubble. There. Oh, got bottom in the camera.
There it is.
All right. You can click in auto heading on Atalanta and bring the heading around to face Hercules. Might tug you a bit. Back up. So this will just take a few minutes while we get situated on the bottom. We have a couple things to check off and some numbers to write down and white balance to do for video. So.
Yep, just let me know when you're ready.
Yep. Well, all right. I want to continue coming down if you want to follow me down.
We already got some good corals and sponges.
Yeah, let's see if I can get something to look at while we...
Yeah, that Regadrella looks nice.
Hey. In the winch.
Contact control, go ahead.
Pilot's in a position where we could potentially come up 30 meters and then go ahead and drop back down 30 meters.
What do you think, Jake? Yeah, we can do that.
Sorry for the delay. We're troubleshooting a mechanical issue with the winch, so we're coming up 30 meters just to diagnose what the problem is.
Roger.
All right. Coming back down.
There's a little fish. It's a Bazazetus.
All right, you can hold there.
Control deck. Yeah, you guys are good to go ahead and continue with dive, but I'll let you know if I need anything else.
All right.
Copy that. Thank you.
All right, resuming white balance.
Sounds good.
Whenever you're ready, video.
Okay, thank you. Okay, it's going to go black for a second. All right, and then do white balance. All right, thank you very much.
All right. All right, ready to go with dive objectives.
Sweet. Our heading will be 015.
015, roger. What was that? Yeah, you can take back gauges. you have a ship move called in, or? Okay.
But we can move, unless we want to look at stuff here, Daniel?
Yeah, I think let's quick snap zooms on some of the big things, but I think, yeah, we should start making our way towards waypoint 2, so yeah, maybe.
All right.
So far I've seen quite a different array of species. We've got unbranched bamboo coral in front of us, and then there's a branching version a little bit further in the field. When we first landed, I saw Ragadrella glass sponge, as well as a Acanthogorgiidae, formerly known as the plexaurids. It's a yellow coral. What else did I see? I saw a Chrysogorgia. I saw Anthomastus. I saw some type of sea star, probably a goniasterid, a cookie star, one of the rocks. So there is quite a bit of diversity down here, and I think it's just going to get more exciting as we go.
I definitely saw all those things too.
Yeah?
Yeah.
Nice. Erin's a professional.
I miss the cookie star. I love those. They're so cute.
It was kind of like on the rocks far off to the back of the field.
Nice.
Yeah.
I gotta fix the sonar.
I think one is Herc USBL and the other one is the DVL. Oh. Okay. Oh, that'll fix it. Oh, thanks. I was wondering about that. Oh, there's that Regadrella. You know, if you go and look down into it, Jake, there's usually some shrimps in there.
Might be able to fly it. Get enough leash to fly over there. Let's see.
Yeah, the cookie stars on that rock off to the sort of upper right.
Ah, yes. You can see it.
Oh yeah, and there's crinoids.
Can come down another 5 meters. Can go for a zoom there.
Yep, on it. Do you want me to go in? Okay, thank you.
There. Yep.
Wow. Oh, yeah, so this is a Sathomatra, a type of feather star. You can tell it from the other ones because it's this nice pretty purple, and it has these really long pinnules at the ends of its arms. Like long filaments. We often see these on corals and sponges, but this one has decided to perch upon a dead piece of coral. And then the sponge is the Regadrella. It's a type of glass sponge in a family Euplectellidae. And inside of the sponge, there's often a pair of shrimps that live in there.
Awesome.
You'll only be able to see them if we look down the funnel. Ready to get going?
Sure.
Yep.
20 meters.
Happy to zoom out.
All right.
Yep, happy to zoom out.
Bridge nav.
Can you put the porch cam back on? The bubble?
Bridge, nav.
Thank you.
We'd like a ship move, 20 meters, 015.3 knots.
So yeah, just to center us back on the dive objectives.
Thank you.
So we have about 12 and a half
Did you say speed?
hours to go, and we have a little over kilometers. So we'll try to balance covering some grounds and getting some good zoom-ins.
Yeah. Zooming on the bamboo coral is helpful to see where it's branching. If it's branching at the nodes or at the internodes. So bamboo corals are called bamboo because they have a proteinaceous part of their skeleton that's very flexible, and then they have a calcareous part of the skeleton that acts kind of like the bones, the structure of the bamboo, allowing it to be both firm and flexible. And the nodes are those little black bands, the flexible bits, and if it's branching at the nodes, we call it a nodal bamboo coral. If it's branching at the internodes, then we call it internodal. So sort of nice way to be able to differentiate some of the bamboos. We're still working on the genetics and taxonomy of this group. It's a very large and very diverse group.
You can go back up a couple meters.
And there's a little sea cucumber as well.
Where's that?
That's right at the bottom of the screen below the stick.
Below the stick.
You've got quite an eye.
Oh, well, you know, it's kind of what they pay me for.
They're getting a good deal.
Yep. So when I'm not here on the ship, I watch this video and I annotate all the animals we see. So it's important for me to spot all these like little things as we go. There might be something interesting on the wood, though.
Yeah.
Yeah.
We can go for a zoom.
All right.
Wood will attract specific species. Not sure what that white thing is. Look like there's like a polychaete, a scale worm on the wood.
I think there's a little urchin on the right side of it as well, just out of view.
Oh. I'm not sure what that white part is. It's an animal. I don't know what it is, though.
There's the urchin.
Oh, yeah. There's a couple of them. Very cool. You just never know until you zoom in a lot of times.
It's true.
That's pretty neat. You see that really thin piece of coral? That might be a black coral of some kind. Oh, yeah. Yep, definitely a black coral. It might be a bathypathes, but a very young one.
That's hot.
That's all the zoom I have.
Okay. Yeah, I'm a bit
great.
far away.
Cool. Thank you.
Want me to pull out?
Yeah, you can pull out.
Yeah, I think if we can start making a little move.
Yep. Atalanta's gonna start moving in a sec. We finished with our ship move.
Is that a fish?
Yeah.
It is a fish.
Where's the fish?
It's in the back over there,
Oh yeah.
just out of the light.
It's probably an eel. There's a little cup coral, and there are a couple of rosellid sponges. A poliopogon, another regadrella.
Front row, do you guys normally like to have the WCA upper cinema cam on view?
Yes, I am waiting on word from Dan that he is done
It's back
messing,
on
messing
now.
with it.
It looks
It's back
like
on.
it's, yeah, it looks like
Okay.
it's on.
We can give it a shot.
Nice. That's helpful.
That's a cool camera.
Yeah.
Well, there's an umbellula.
Perfect.
Those are Gabby's favorite. And then there's this really cool crinoid, a really long Proisocrinus ruborimus. The common name is sea lily.
Yeah, the stereo cam seems a bit laggy.
Little sea cucumber,
Can you turn on
probably
downlight
amperima or
for me?
peniagony.
Downlight? Should be on Herc page.
There's a
Hotel.
Culeolus as well.
Lights. Lights. And then down. You can turn it on.
Nice.
That's what I wanted.
Oh, there's a little holotherian.
Yep.
Do you want me to zoom in at all?
Yeah, you can go for a little zoom.
It's a little elpidiid, either peniagony or amperima. And this is a feather star called Proisocrinus ruborimus. And on its stalk you can see some hydroids. You often see this association of hydroids on the stalks of these feathers or sea lilies. And sometimes
All right.
the crinoids
Come
and
on,
feather
let's
stars
do
will
that.
have associate snails that live
Think
on
of
them. So it's always a fun treat to zoom in and see if that snail is there. There's also a sea pen, two different species of sea pen. There's a umbellula, and then there's also a rock pen over on the right side. And then there is a tunicate called Culeolus. It's on a long stop right on that rock there, that little floaty bubble. Pretty neat, huh?
Do we want to call in continuous ship moves?
Yep,
Or?
I'm gonna keep us moving if that's good
All right.
with you.
Yep, it's good with me.
Bridge nav. We would like another 20-meter move, 015.3 knots. Thank you. can we look at that little puff ball on the top of the rock?
Sure. Yeah, I don't know what's going on with the WCA cameras. They have been power cycling randomly, so.
Is it the lower puff ball or the?
The upper puff ball. Yeah, so we have two different type of tuna kits on the lower, and then on the upper it was...
up higher. That's a tuna kit.
Yeah, we can get some good look at that tuna kit.
That one?
Yeah, that's the one.
I thought the other one looked more like a puffball. That was just me.
That one looks kind of fuzzy and puffy to me.
Yeah. Yeah.
Did you want to zoom on this one?
Yeah, that'd be great.
Let me know when you're ready, Jacob.
Okay, 1 second.
I think it's a type of Chrysogorgia.
All right, I can go for zoom there.
Yeah, definitely Chrysogorgia. And there's a bunch of little demisponges on the rock as well as a snail. Very nice. So I think this is Chrysogorgia stellata. All right, that's good for me. That's good for you.
Good for me.
Yeah.
Good for me.
Sweet.
Hey Megan, there is a question about the puffball.
Uh-huh.
And so what is the approximate size of the fuzzy puffball?
So if we turn our lasers on, we have scaling lasers that are 10 centimeters or 4 inches apart. And I believe, based on my experience of this camera in particular, that size was about 4 inches.
Thank you.
You can see the lasers on the first satellite feed now.
Yep. Yeah, that's really, the lasers are very, very helpful because a lot of times when we zoom in, things appear a lot larger than they are in real life. And then we're zoomed out or flying particularly high, things might seem quite small, but in real life they're quite large. So the lasers help keep our frame of reference and understanding the size of these organisms. And the scaling lasers can be used for a number of different analyses after the fact to better understand size distributions of different types of corals and sponges and other animals.
There's something floating in the water there.
Bridge nav. Yeah, we'd like to move 20 meters, 015. Thanks. Daniel, do we have any objectives for collecting a rock sample at the seamount?
Yeah, you kind of read my mind a little bit, but I have not. So definitely we should try to get one. Loose angular rock. That's a nice view there.
Yeah, yeah. It's quite a bit of diversity. We've got our Iridogorgia. There's some polyopicon sponges, some chrysogorgid corals, bamboo corals. Kind of interested in that coral that's the sponge that's hiding behind the rock there. But of course, we should probably look at the Iridogorgia. It's a favorite.
It's just so cool.
It's just Iridogorgeous.
Gosh.
I say this every time. People who know me will be like, ah, Megan. So this might be Iridogorgia bella. It has a tighter whorl, and the branches are concentrated at the top of the animal. I think there's a shrimp in there too. Might be good to get a zoom to see what kind of shrimp that is.
You can go for a quick zoom.
I think it's a Bathypaymonella. It's a type of shrimp that's in the Carididae. They have these like really nice long front pincer arms. Yeah, definitely Bathypaymonella. Oh, that's great imagery. I like these ones because, oh, this one is a female. You can see she's got her eggs, holding them in her pleopods. They're the bright purple. And we often see these shrimps associated with this type of coral. So it's nice to keep seeing these same associations. We're kind of building a bigger picture of what the communities down here look like.
You want me to pull out?
Yeah, you can pull out a little
Yeah.
bit. Yeah.
Okay.
Yeah. I think that's the unofficial common name for this coral is the firework coral. Yeah. You're not the first person to say that. Ooh, look at that Calyptrophora.
Good little ridge right here.
Yeah.
Nice current.
And it's probably nice all the time. Look at how many corals we're seeing. They like it here.
Yeah, and quite diverse.
I really like these Candelabra Calyptrophora corals. They're really pretty.
Tempted to fly right through, but
Just like
I know it won't end well.
they get bigger. Maybe they'll be a big enough one. Yeah, and these unbranched bamboos, they can get to be like 3 or 4 meters long. They're just really impressive. Getting the Jake special, doing the nice pirouettes.
Beautiful.
You see this white coral right there, the little one?
Tucked away in the rocks?
Yeah. I'm not sure what that one is, if we could zoom it. Because it's not a bamboo like the other ones around it.
These lights are all messed up. All right, you can go for a zoom there.
What are you?
Sorry.
I think it's a hydrocoral.
Happy for me to push in more?
Yep.
Cool.
Or it's an Anelopsamia. Yeah.
That's a sclerotinia.
Yeah, it's sclerotinian. Very cool.
It's like a skeleton.
It's still alive.
Very Halloween-y.
So these are deep sea reef building corals.
All right.
Yeah, it was quite small. Oh, is that a cumation? You see that little
Oh, that little
like
floating around?
floating thing? Yeah, it's a type of crustacean called a cumation. They're very, very small, and you don't usually see them on camera because they are so small.
Okay.
Very neat.
There's some sort of crab on there?
Yeah, it looks like a hermit crab. You see it's in its
Oh,
shell,
yeah.
and then there's a hydroid on the coral as well.
Pull out?
Yep.
Yeah, cool. Magic. I love zooms. They always like reveal so many cool things.
I love them too.
Oh, the corals in the crevice.
Yeah, the current must accelerate like down through there. That rock might be loose, you think? What do you think, Daniel? Like in between these two little pieces of rock, there seems to be one that might be a angular one. Yeah. This might be a good area.
Collect our first
Yeah,
sample.
that's
Collect our first sample and call it a watch, and then come back for dinner relief.
yeah. I think that if that's in a good spot.
What do you think, Jake?
I think we could grab it.
Okay.
That's annoying.
So we'll use these rocks in order to understand a little bit more about the geology of this area, the origin of the seamount, how old it is.
Yeah, ideally, we would like to get a rock that's still like partly attached, so we know that it came from that exact spot. I mean, these probably just crumbled right off.
Yeah.
So I think these are going to be fine, but if we see something that looks like it's mostly cracked but not fully off the main outcrop,
Okay.
that would probably be a little bit preferred.
Yeah.
Advice to move on.
Yeah, we can keep looking
Than just
around.
a loose one.
I feel like
Yeah.
this seems like a good area for potential.
Definitely. It's
samples.
hard to tell how weathered these are if you were to grab one on kind of one of those little edges and twisted it, if it would just crack off, or I think.
This area seems pretty well manganese crusted, so it might not break off easily.
Yeah, I agree. It's very cemented.
Definitely. Wow, look at this.
That's really pretty.
Must be a good current flow there.
It must be.
Nice stauropathes, Evercrassogorgids, bamboo corals. Yeah, my favorite fact about these are that they're kind of like windmills, and they'll adjust where they place each other depending on where the current is flowing, they can just change direction.
And you see a little squat lobster in the Chrysogorgiid. It's gorgeous. Well, it's definitely a highly productive area.
Looks like we're 5 minutes to watch change, so we'll explore this area a little bit until the next shift comes on.
While we're waiting on shift change, your relief pilot, can we take a look at the brown coral to the bottom right?
Sure. The one right here? Yep.
Yep.
Yeah, let's get up nice and close.
Well, welcome everyone to our first dive of the expedition. This is expedition NA167, or Lebu's voyage, as we're exploring the Palau National Marine Sanctuary. We're about 45 miles north of Vasco Reef, which is kind of the northern end of the main group of islands in Palau. And we're just going to hang out here for a moment while we finish up watch change in the van.
Hercules. And now you're unmuted.
Hey, Dan, do you have your headset on?
Still doing a handover.
Oh, okay. I just wanted to push a button over here, and I'm afraid it might do something to their screens, and so I want to warn them. Okay. Cool.
Hello, everyone. My name is Elle, and I just changed shifts, so we are gonna start with one Palauan word of the day, which is Ali. So Ali, to everyone who's tuning in, my name is Elle, and I am a Science Communications Fellow on this voyage. And if the back row is ready for introductions, could I have the back row start?
Hey, yeah, I can go ahead. Thanks, Al. Hi, everyone. I'm Brian Kennedy. I'm one of the co-science leads for this expedition, and I'll be the 4 to 8 watch lead. I'm a deep-sea benthic ecologist, and my research interest generally is large-scale, ecology process between benthic fauna and seamounts in the Pacific. But I'm probably a little bit too curious for my own good and don't narrowly focus on anything other than that kind of broad category. And I'll be helping guide us on the 4 to 8 as we do some of this really important exploration here in the Palau National Marine Sanctuary. I'll turn it over to Harold next.
Thanks, Brian. My name is Harold. I'm a second-year PhD student at the University of Hawaii at Manoa, and I study deep-sea ecology as well. I mainly focus on deep-sea habitats, coral gardens, sponge gardens, and how they relate to fishes. So I'm curious to find out how fish use different habitats like these in the deep sea and what mechanisms link them. So I'm interested in food webs, reproduction, spatial habitat use, also very broad interests like Brian. So I'm excited to be in the science chair from 4 to 8 on this cruise.
Hi everyone, I'm Kayla. I am the Ocean Science Intern aboard NA-167. For today's dive, I'll be the data logger, and I'm currently being trained by the amazing May. And for the next 4 hours, I'm excited to see what's in store for us. Oh, yeah.
Thank you, back row. And to say thank you in Palauan is Sulang. So Sulang, back row. Okay, we have one more in the back row. Could you please introduce yourself?
All right, can everybody hear me?
Yep.
All right, my name is Jeff Drazen. I'm a professor in the oceanography department at the University of Hawaii, and I am also a deep sea ecologist. And in particular, I enjoy studying the ecology of deep sea fishes. And it's really exciting to be here on this expedition. I'm actually in the room before my shift actually begins, which is at 8 o'clock. Thanks.
Sulang, Jeff.
So we're just hanging out here for a bit. We've got a couple new people to the team, and this is their first shift, and so we're doing a detailed hand passed on and training session here in the front row, and then we'll get started with our exploration here very shortly. So for the next few minutes, just enjoy the scenes of the seafloor, and then we'll get our way on up this feature. One of the cool things that I really enjoy working on Nautilus is how much of a training vessel it is. We have multiple interns. We've got several people, this is their first or second time going to sea. So we're always training and bringing up the next generation. And so it's a real honor to be part of that, and I enjoy the enthusiasm that all the newbies bring to the vote.
So I'd like to give some updates for this dive plan. Currently, we are 45 nautical miles north of the edge of Velasco Reef. And for Palauans that are listening, Velasco Reef is part of Kayang. Kayang is our northernmost state. Palau has 16 states. So yeah, right now we are 45 nautical miles north, and we are
talking on SPL. Good afternoon, everyone. They're ignoring me, so.
Good afternoon, pilot.
Good afternoon, Dan. How's it going?
It's going good. Thank you for your patience there. Just Jaden's first time in the front row here, so
absolutely
going through my little spiel.
Well,
meanwhile, I seem to have gotten lost. I don't know. I was not paying attention to what the ROV was doing.
Well, we have done a relatively good job of documenting this little outcrop of coral, and so I think we are ready to get on our way up towards waypoint 2.
Roger. I'm gonna stand by one. So you see, Jaden, now as I turned my heading and your heading got pulled around, and you can see on your blue box there, the one we talked about Hercules, but we never did talk about it, Milana. So what I'm doing is I'm what we call I'm pulling on your leash. So as I turned, the vehicles were facing away, and it pulled Atalanta around, and because you have auto heading on, and now that I've gotten closer to you, Atalanta corrected itself. It won't be long before you're yelling at me when I do that, because it is very annoying. Okay, I think I'm back in the neighborhood of where I was when I sat down here. We drifted 10 or 15 meters to the southwest there while I was yapping. Zero 20. Oh, I'll just keep going this way then. Roger. Hey Mike, if you're listening down there in the data lab, Amber's fancy NDI encoder is choking.
Is it only mine when it's being buggy?
I'm gonna try and cycle the NDI on that camera, see if that. But I don't think it's my camera. I think it's your encoder.
It's ours. It's a collective.
Our encoder.
In the handover, they mentioned that they had cycled it a couple times.
And that didn't help. I imagine it's the not bird dog, the better one. The better one. It always buys the good stuff. So that was a bonk. Contact. Watch out for that giant rock. Okay, Chris, I think I'm ready if you want to
start moving.
Sure. Take it nice and slow because it's been a while since I've operated an ROV. I forgot where all the buttons.
Yeah, understood.
Controls are.
Bridge nav. Could we do 20 meters 020?
So in general, Jaden,